Hello, everyone. My name is Corey Arnold, and I'm an associate professor at UCLA in the departments of Radiology, Pathology, and Bioengineering, and I also direct the Computational Diagnostics Lab. In this talk, I will be discussing some of the work that our lab does in image analysis in stroke and prostate cancer. So we do a wide variety of work within the lab, including mobile health work and heart failure, and a lot of EHR data mining. But as I said, in this talk, we'll focus more on our image analysis work. At UCLA, we do a lot of ultrasound fusion targeted prostate biopsies. This procedure generates an interesting data set that I've attempted to show on the top right here, where you see the contour of a prostate rotating. Within the prostate, you see several additional contours. These were drawn by the radiologist and delineate areas that are suspicious for cancer. And so these ROIs are used during the biopsy procedure in that the images are fused in real time with ultrasound. You can see the lines through the prostate. These represent biopsy cores that were actually taken from the prostate. And so this project attempted to utilize this interesting data set, and we had the hypothesis that we could build a model to predict whether a prostate biopsy core will contain significant cancer using our MPMRI data alone. So we took our T2, ADC, KEP, and K-trans sequences and the associated biopsy ROI data from 675 patients. We had a total of around 9,000 cores, and we built a rectangular solid image region that we generated by interpolating a series of perpendicular planes to the biopsy needle. And this is shown on the lower right here, in which we can see an imaging core on T2, ADC, VVAL, and K-trans. And so we generated features from these. We generated 158 features, and we trained in SVM. So no deep learning was used in this work. And we achieved an AUC on our test set of 0.72 with a precision of 0.87 and a recall of 0.74. And we presented this work a few years ago at NeurIPS. And so that was an interesting project, but it really leaves voxel information on the table, right? We want to use all the voxels within an image to try to detect prostate cancer. So we subsequently used the ROIs generated by the radiologist to train a model to automatically segment areas suspicious for prostate cancer based on PIRAD scoring. And PIRAD is the standardized scoring metric used by our abdominal radiologists to specify the suspicion of prostate cancer, okay? So here our methods included using the T2 ADC calculated high BVAL images and our PIRADS ROI annotations from around 1,000 patients. So each patient had the MR data, had the contour, and each contour had a PIRAD score that we used as the target for training the model. We used a UNet, a 3D UNet specifically, and we trained it with a 128 by 128 central column. So that's where the prostate always occurs in our image sequences. And we used a focal binary cross entropy loss. And so we had to do this because of the imbalance in the voxel data, right? Most of the voxels are not cancer, only a small amount are. And so we need to focus our loss on that region. You can see our results. We had an AUC of 0.95, which is misleading given the imbalance in the data. That's reflected in our average precision and our precision at 80% recall scores. Nonetheless, I've shown two examples here in the left column where the blue region is the ground truth and the red is what we predicted. And you can see that the salient region of our predictions does correspond with the ground truth. So we're now in the process of trying to refine this model to eliminate a lot of these false positives. So a brief aside, I hope I've described this data set in enough detail so that you can appreciate other things we can do, including looking at where we find cancer within the prostate relative to the ROI specified by the radiologist. So because we know the coordinates of every biopsy core and we know the ROI, we can see how far away do we detect cancer from an ROI? And that's what this graph shows here. So on the x-axis, we see distance from the ROI surface, the ROI here being the region within the prostate that the radiologist thinks is cancer. And on the y-axis, we see the cumulative probability of detecting cancer. So, for example, once we're 10 millimeters away from the ROI, we've already detected over 80% of our grade group 4 and higher cancers, right? So the intuition here is cancer tends to occur close to ROIs delineated by our radiologist, and that simply sampling around that region, which we call the penumbra, you can detect pretty much all cancer and reduce the total number of biopsies needed. And so that's shown in this table here, where you can see once you move 15 millimeters away from an ROI surface, we've detected 97% of all cancer, and we can reduce the number of biopsy cores taken by over 5. These two graphs at the bottom just illustrate this notion of a penumbra, where on the left we have what was drawn by the radiologist here. On the right, we have an expanded penumbra. And our premise for this paper that we just recently published in the Journal of Urology is that one might considering just sampling the penumbra region rather than doing a template biopsy procedure. So moving back to our deep learning work, we recently had a collaboration with NVIDIA, the National Cancer Institute, and SUNY Upstate in New York on prostate segmentation. And the goal of this collaboration was to study federated learning. And so prostate segmentation is a pretty straightforward problem. There are already good algorithms for it. I've shown an example here of a ground truth with a predicted region. You can see it's quite good. And the goal of using this as a task was really to minimize any concerns about having to share intellectual property if we discovered a state-of-the-art algorithm, and also because there's lots of data available to do it. And so we took 100 T2 weighted prostate MRIs from each institution. So we had a total of 300. The MRs were using local clinical protocols for prostate cancer diagnosis. Each MRI was annotated with a prostate contour by a specialist radiologist. And then each set was divided into 80 training MRIs and 20 held out test MRIs. And we then grabbed 343 additional T2 weighted prostate MRIs from the Prostate X challenge to use as an outside test set. So for those who aren't familiar with federated learning, how it works is data stays within sites within the federation and the model moves outside. And so there's this federated server that contains a global model. The server pushes that model to each site in the federation. The site trains on their local data, in a sense, updating the model. It then pushes that model back to the federated server, and all the models are aggregated and then sent back out. And so this process iterates until some convergence criteria, but it allows each site in the federation to indirectly benefit from data outside of their site. So these are the results from our project in prostate segmentation. What you can see in this first table, the first 3 rows are private models. So these are models trained only on site-specific data. And we see how they perform at data from their site as well as data from the other sites. And in general, what you see is along the diagonal, these models perform very well because they're trained on their data and they're tested on their data, but they perform more poorly off diagonal, right? So when they're trained only on their data, but tested on outside data. Whereas you can see the last row here, the federated model performs better in general on data from all sites. Okay, so it's more robust. We see that again on this table on the lower right, which was with the Prostate X data set. So this was data that was not used for training any model. And we see that the federated model performs better than any model trained at an individual site. And we recently had this published in JAMA if you're interested in more details. So moving on to histology analysis, what I've shown here is a lung wedge resection. It's around 5.5 billion pixels. To give you a sense, here's what it looks like at full resolution. An entire prostate T2 sequence could fit within this patch. The entire CFAR10 data set could fit within this patch. And we recently acquired a Leica GT450 scanner, and so we can generate one of these every roughly 32 seconds. And so we're generating a lot of data here and paying for a lot of storage. So in this project, we have all of the histology cores from all of our prostate biopsy procedures, and we sought to develop a deep learning algorithm that can automatically discriminate cancer and Gleason grade on histology using only core level labels. So we have clinical prostate biopsies from around 830 patients, and they had around 20,000 biopsy slides. So in terms of annotations, we had a couple decisions to make. Did we want to try to generate pixel-wise annotations, which are very expensive and challenging to do with this volume of pixel data? There's also coarse contour annotations that we could use, in which the pathologist could outline a region on histology and label the entire region. And we could assume that every patch within that region had the same label. Or could we use slide level annotations where an entire slide is assigned a Gleason grade? We don't really know where within the tissue that grade corresponds, but we know it's in there somewhere. And we have tons of these things. And so we decided to use slide level labels because we have so much data, we didn't have to get any additional annotations. And we used a framework called multi-instance learning. Briefly, in a supervised scenario, each sample has its own label, right? So we have triangles, and triangles have labels based on their color. In a multi-instance learning scenario, we have bags of samples, and each bag has an associated label. So here you can see a bag has a label of 1 if it contains any of these kind of greenish triangles. Otherwise, it has a label of 0. Okay. So this is analogous to what we have in histology, where we have a slide. So this slide has a label of 1, meaning it contains malignant cancer. We can tile this slide because it's so big, and then we could consider this a bag of tiles, and this whole bag would have a label of 1. But for an individual tile, we don't know the label, right? And so a multi-instance framework allows us to use data in this format to actually learn to classify slide level labels. So this was the algorithm we developed. The technical specifics are sort of beyond the scope of this talk. What's worth mentioning, though, is that it's a multi-scale model, so it operates at 2 magnifications. So at the first magnification at 5x, we feed in tiles, and we have a CNN backbone that extracts features. We combine those features with an attention module to simply detect cancer. Then for tiles that we think contain cancer, we send those on, we remagnify them to 10x, we extract features again, and then we do grading on those tiles. And again, we have an attention module here. So for more details, you can look at our recent publications on this. So here are some examples. So again, what I'm showing here are tiles with their associated attention values. Because we don't have tile level labels, we don't know if these are right or wrong. So we compare them to the marker lines that our pathologists draw during interpretation. And you can see that our attention values seem to correspond quite well with what the pathologist was looking at. So how does this model perform? This table shows various different versions of the model with the proposed version at the bottom. You can see for cancer detection, we have an AUROC of 98 and an average precision of 97. So I would argue it works pretty well. It certainly, in my opinion, works well enough for clinical use. For cancer grading, I have this confusion matrix here, and you can see that it does very well on benign, pretty well on grade group 1, and then performance falls off a bit for these other grade groups. That's a function of a couple things. One, we don't have as much data, for example, in grade groups 4 and 5. And also we see more inter-rater disagreement among pathologists in these areas. So it could be that we have labels that are sort of noisy. We do see, though, that when the model fails, it's usually only off by 1 grade. So that's nice to see. Briefly, I wanted to touch on our integrated reporting project at UCLA. So at UCLA, we do integrated radiology and pathology diagnosis for cancer, or integrated reporting for cancer diagnosis. I've shown an example report here where we just have the patient's sort of clinical history. And then on the assessment tab, we bring together the radiology and the pathology results. And so here's the data I discussed previously from our targeted biopsy procedures. Here's the corresponding pathology report for this patient. And so the goal of this report is to bring the most salient information to the fore to allow the clinician to quickly get up to speed in a patient's case and maybe even share the data directly with the patient. And it also gives us a platform for beginning to integrate some of our machine learning results into practice. And so this is a beta version we have running in which we have a histology quantification application. So our deep learning algorithm for quantifying Gleason grades runs sort of in the background. We have this 3D render of the prostate where the user can come in and rotate it and zoom in and out. And then we have a population map. So here we took a bunch of patients, registered their prostates together, and created a heat map for where we see cancer at UCLA. And this allows a user to sort of contextualize for a given patient how they relate to the rest of the UCLA population. So shifting gears to our stroke work briefly. Our main project here is in stroke onset time classification. And our hypothesis is that MR signal contains latent information that can be used to classify times in stroke. And this has an implication for treating patients that arrive with unknown stroke onset time. So that's roughly 20% of people in our ER who show up, either they're unconscious or they don't know when a stroke occurred, and they're not eligible for one of the best treatments in stroke, which is a thrombolytic therapy. So at UCLA, we took around 350 patients. We also have a data set from collaborators in South Korea. What we did was we trained a slice-wise deep learning model. So we took our DWI, FLAIR, and ADC sequences, and for each slice, we trained a model. It first generates features with a CNN backbone. We have an attention module here. There's one backbone per slice, and we then append the attention and convolutional outputs at the end, and we have this trainable slice weight W. So we sort of sum the features, the weighted features from each slice together to then predict stroke onset time. And there's many more details in our recent publication in CMIG if you are interested. We train this model in two phases. In the first phase, we just do stroke detection. So this is kind of pre-training in which we feed in halves of the brain, affected and unaffected, and we train the model to detect affected halves of the brain, stroke affected halves. We then freeze the first two convolutional layers, and we feed in only affected halves to classify times in stroke, okay? So we also had 3 radiologists review the images for DWI FLAIR mismatch. This is a current kind of biomarker for measuring stroke onset times, and we used it as a label as well here. And what you can see in this sort of busy table is that we trained models on internal data, external data, and both data sets. And what we found was the best performing model was trained on both data sets. We can evaluate it on held out test sets from UCLA, as well as from AMC in South Korea. And we find that those models perform the best with about a 0.8 to 0.823 AUC. Using mismatch as a target, our best model got about a 0.75 AUC. But it's important to note that mismatch is a difficult thing to assess even for radiologists, in which their kappa values range 0.37 to 0.63. So here are some attention maps showing where our algorithms focused their attention for making these classifications. So there's 4 different cases here. I'm just going to go over a couple. This first case here is under 4.5 hours, which is a treatment threshold that we used. So the patient is 79. The radiologist thought that there was mismatch, and we predicted under 4.5 hours, which was our binarized threshold for this problem. The other interesting case here, I think, is this patient is 4.5 hours, so just over the treatment window. The radiologists all felt that there was mismatch, indicating that this was a young or an early stroke. And our model also predicted that the patient was under 4.5 hours, which, although it conflicted with the ground truth, it agreed more with the radiologists, and we think reflects what's more important here, which is a tissue clock rather than actually a wall clock for dictating therapy. So thank you for paying attention to this brief talk. I wanted to acknowledge everyone on our team, including the faculty, staff, and students and postdocs that did this work, as well as our funding sources. Please email me or go to our website if you have any questions.